Residual host cell proteins in DNA are host cell related impurities that we have to control for during biological manufacturing processes. They're not the product we're looking for. There's something extra that we have to try and remove by the processes we develop.
So they are actually they're generated from the host cells because of the recombinant technology we use to produce biologics or therapeutic proteins. Normally we use mammalian cells such as Chinese hamster ovary cells, CHO, or we use E. coli bacteria cells or some other hosts. So they are whatever endogenous proteins or DNA that present in these host cells.
There is the thought that host cell proteins and other impurities similar to that can act similar to adjuvants and actually cause immunological responses. And if you have that kind of response take place, you could have antibodies against the drug that you're giving, or you can even raise antibodies against the endogenous molecule in the body. So you want to try and make sure that these products are safe and efficacious, and you don't have neutralizing antibodies or anything like that that would cause an adverse event.
The technologies for detecting the host cell proteins and DNA, they're actually quite different because there are different challenges associated with the both. And with the host cell DNA, it's more straightforward just because that DNA is in the background of a protein, so it's relatively easy to separate the two.
Well, with host cell proteins, typically it's an immunological reaction or immunological assay. That's because there's such a broad spectrum of host cell proteins that could be in there. You need to use a broad range of antibodies in order to detect and quantitate those materials that are in there. As far as residual DNA, there's a number of techniques out there, but one of the more popular ones happens to be the polymerase chain reaction technology, sometimes called PCR, and those are some of the current technologies that are being used.
I am on the expert panel for the DNA measurement, so we are actually developing the reference standard for whole cell DNA. So this could be potentially used or referenced in a, say, a product monograph. Ideally, it would be nice to have also a reference standard for whole cell proteins, but that could be a challenge because it depends on what cell line you use, and there are so many different cell lines people are using for mammalian cells, and then what kind of techniques you use to recover the proteins from these cell lines. So there will be discussions on these aspects.
A lot of companies know how they go about measuring these impurities, but the value of USP is to come together and give some general guidance that all companies can look at and understand how to measure these quality attributes. You know, some of the bigger companies have the capabilities to go out and make some of the critical reagents for themselves. Some smaller companies that are trying to develop these kind of molecules, they may not have those kind of resources or may have to contract that sort of thing out. The advantage that the USP does is it puts together expectations and procedures and standards that allow people to, or companies to develop these assays, and with reasonable certainty that what they're doing will meet the regulatory hurdles. People who are involved in writing the guidances and are involved with developing the techniques are all going to be here discussing what the future is going to look like. So you may have some procedures in place that you're following, but it's always good to benchmark what the others are doing and get a sense of where this kind of technology is going in the future. That's what's going to be discussed in the meeting, and I think that's what has the greatest potential and the benefit to people in the biopharmaceutical industry.